So let's look at genomes. And for further information, as always, you could go to the genomes or genomics or proteomics. Yeah, where proteomics is the study of the basically the proteins or set of proteins of an organism. So going further, so a genome is an organism's complete set of DNA, including all of its genes. Yeah, so sequencing and analysis of genomes can be done using high throughput DNA sequencing and bioinformatics. Bioinformatics to assemble and analyze the function and structure of an entire genome. MES note, in general terms, throughput is the rate of production or the rate at which something is processed. And DNA sequencing is a process of determining the nucleic acid sequence, the order of nucleotides in DNA. It includes any method or technology that is used to determine the order of the 4 bases adenine, guanine, cytosine, and thymine. Let's continue further. Here is an example. Let's make it a bit smaller. Let's make it smaller. So an example of the results of automated chain termination DNA sequencing. So basically, just a computer algorithm / molecular technology to determine all of the base pairs or the molecules there. So you have ATTG, or not just base pairs, I mean the order of the nucleotides. So you get a strand of DNA or strand of RNA, and then you get the full sequence. So A, T, G, C, T, T, G, and etc. And you can do other methods of determining. And you can see the waveform here. That might be doing some sort of electrical signaling as well. So there's high line there. That's for the T. There's the green ones there. So green for the A. So anyways, continue further. So bioinformatics, bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data, in particular when the data sets are large and complex. And now here's an example of early bioinformatics, which is a computational alignment of experimentally determined sequences of a class of related proteins. Yes, you get an amino strand sequence of proteins, and basically, you line up a whole bunch of different proteins, and you line up where the molecules match up, or the amino acids, and so on. Fascinating stuff. Yes, that was an early setup, and now here's the newer ones. It has some a lot of information and also some graphical representation. So, map of the human X chromosome from the National Center for Biotechnology Information website. So there's a chromosome there. There's the I believe the centromere there. And there is a whole bunch of other information here, as well as there's some genes right here, gene sequence, and symbols there. We click it and we'll show you the full thing and so on. It's fascinating stuff. I'm not going to go into that. I just wanted to show an illustration of it. And this would likely require a video on its own just to analyze all that. And this is this right here is an ideogram, or ideogram. Let's just continue further. So an ideogram or ideograph is a graphical symbol that represents an idea or concept or specific words and phrases. So this is basically a symbol that represents different sequences and genes and so on. Gene mapping. This is a map of human X chromosome. Gene mapping describes the methods used to identify the locus of a gene and the distances between genes. Gene mapping can also describe the distances between different sites within a gene. And let's go over this locus over here. So in genetics, a locus or plural loci is a specific fixed position on a chromosome where a particular gene or genetic marker is located. And also, I just googled the pronunciation for this. It's actually, Google says it's loci. So I was calling it loci or loci, but it's actually loci apparently. Yeah, so basically it's a fixed position on a chromosome where a particular gene or genetic marker is located. Going further, so the essence of all genome mapping is to place a collection of molecular markers onto their respective positions on the genome. Molecular markers come in all forms. Genes can be viewed as one special type of genetic marker in the construction of genome maps and mapped the same way as any other markers. There are two distinctive types of quote maps used in the field of genome mapping: genetic maps and physical maps. While both maps are a collection of genetic markers and gene loci or loci, genetic maps distances are based on the genetic linkage information, while physical maps use actual physical distances usually measured in number of base pairs, which is pretty fascinating. Let's go continue further. So a genetic marker is a gene or DNA sequence with a known location on a chromosome that could be used to identify individuals or species. And here's the genomic map of chicken. And there's a whole bunch of chromosome 1, 2, and etc. And then every single gene on there that it has been known, and so on. Fascinating stuff. Yeah, there's a couple little chicklets. So chicks on top of a picture of a genetic map of a chicken. The chicken genome has 39 pairs of chromosomes, whereas a human genome contains 23 pairs. Yes, interesting. Yeah, so it has more chromosomes than humans. Very fascinating. So a molecular marker, and this is because we mentioned it here. So molecular markers, the essence of all genome mapping is to place a collection of molecular markers onto their respective positions on the genome. So a molecular marker is a molecule contained within a sample taken from an organism called a biological marker, or it's a type of biological marker, or other matter. It can be used to reveal certain characteristics about this respective source. DNA, for example, is a molecular marker containing information about genetic disorders and the evolutionary history of life. Fascinating stuff. Let's continue further. Now let's look at here. So many genes encode more than one protein, with post-translational modifications increasing the diversity of proteins within a cell. A cell's proteome, or proteome, is its entire set of proteins expressed by its genome. So here's a diagram of the different organizational levels. So MES note a general schema or framework showing the relationships of the genome, transcriptome, proteome, and metabolome, or the lipidome, in the total interactome of a cell. And now they're getting a bit carried away with themselves. So we have genome is all the set of DNA. Transcriptome is all of the set of RNA. Then you have proteome or proteome, proteome as a set of all proteins. Then you have the metabolome as the set of all sugars, nucleotides, amino acids, and lipids. Yeah, and the lipids has its own lipidome inside there, and this direction here, you can include proteins in its lipidome, as well as RNA could be all inside there. Yeah, because lipids usually just a variety of a bunch of stuff together. And yeah, we could just go refresher on lipids, go over here. Generally, let's go over here. Let's look at this one right here. So fat may also be used more broadly known as a lipid, which is any substance of biological relevance composed of hydrogen, carbon, hydrogen, or oxygen that is insoluble in water but soluble in nonpolar solvents. Although lipids are defined more generally than fats, thus making them a subgroup of thus making fats a subgroup of lipids. And here I just teleported to the other part when we discuss lipids. So lipids, unlike the other macromolecules, lipids are not defined by chemical structure. Lipids are any organic nonpolar molecules. Yes, it can include basically anything in there. And they are, some are huge aggregates of small molecules held together by hydrophobic interactions. So just a bunch of stuff together, and they are only class of macromolecules that are not made up of polymers. And the most biologically important lipids are fats, phospholipids, and steroids. Alright, so we're back here, and yes, we're back over here. Let's just center this right here. And all these together, these sugars, nucleotides, amino acids, lipids, are known as metabolites, and then they are, then after that, they make up the molecular machinery for the phenotype or function of the organism. And so this interactome is the whole set of molecular interactions in a particular cell. And the transcriptome is going to basically go over these directly, and just the definitions is pretty straightforward. So the transcriptome is a set of all RNA transcripts, including coding and non-coding in an individual or a population of cells. A metabolite is or metabolite is an intermediate or end product of metabolism. The term metabolite is usually used for small molecules, and the metabolome refers to the complete set of small molecule chemicals found within a biological sample. Yeah, such as here. So you have nucleotides, amino acids, sugars, etc. Especially if you have them all over the place, it's an end product. And going over here, so the lipidome, it refers to the totality of lipids. And going further, so the genomes of prokaryotes are small, compact, and diverse. In contrast, the genomes of eukaryotes are larger and more complex, such as having more regulatory sequences, and much of its genome genome are made up of non-coding DNA sequences for functional RNA, so this one right here, ribosomal RNA, transfer RNA, tRNA, and messenger RNA. Yeah, although I'm not sure why this mRNA is there. It's functional RNA, so this non-coding DNA sequences for functional RNA, but I believe this is messenger RNA still going to be coding. But yeah, you get the general idea of this sentence from Wikipedia. So basically made up of non-coding DNA sequences for functional RNA or regulatory sequences. Going further, so MES note, a regulatory sequence is a segment of a nucleic acid molecule which is capable of increasing or decreasing the expression of specific genes within an organism. Yeah, so it basically regulates the expression of the genes. Yeah, going further right here, a non-coding RNA or ncRNAs is an RNA molecule that is not translated into a protein. The DNA sequence from which a functional non-coding RNA is transcribed is often called an RNA gene. Yeah, I discussed this earlier. Let's keep going with it. Abundant and functionally important types of non-coding RNAs include transfer RNAs or tRNAs and ribosomal RNAs or rRNAs, and these are primary component of ribosomes. Yeah, as well as small RNAs such as microRNAs or miRNA, siRNA, which is small interfering RNA, piRNAs or Pee-wee interacting RNA. Yeah, which is again a small RNA, so Pee-wee interacting RNA. And then this one, SNO-RNA or SNO-RNAs are small nucleolar, nucleolar RNA. So yeah, again, another type of small RNA. Then you have snRNA, small nuclear RNAs, and xnRNAs, extracellular RNA, scRNAs, small caudal body specific RNA, and the long ncRNA such as this one here, XIST, and hot air. Yeah, I'm not going to go over too much of those. I just wanted to illustrate the different types over there, and they're each one of their different terminology and so on. So the number of non-coding RNAs within the human genome is unknown. However, recent transcriptomic and bioinformatic studies suggest that there are thousands of them. So yeah, thousands of these types there. The total number of these RNAs, yeah. Many of the newly identified NC nuclear, or yeah, this is one right here, NC non-coding RNAs have not been validated for their function. It is also likely that many ncRNAs or non-functional RNAs many non-coding RNAs are non-functional, sometimes referred to as junk RNA. And there is the common the common or the infamous phrase junk RNA or junk DNA, and are the product of a spurious transcription. The spurious transcription, I just want to put here definition as dubious or unnecessary. Yeah, or random, or etc. Other terminology you could use to describe the spurious definition. So non-coding RNAs are thought to contribute to diseases including cancer and Alzheimer's. All right, continuing further. So Alzheimer's disease, AD, is a neurodegenerative disease that usually starts slowly and progressively worsens. It is the cause of 60 to 70% of cases of dementia. The most common early symptom is difficulty in remembering recent events. Neurodegeneration is the progressive loss of structure or function of neurons, which may ultimately involve cell death. Continue further. So dementia manifests as a set of related symptoms which usually surface when the brain is damaged by injury or disease. The symptoms involve progressive impairments to memory, thinking, and behavior, which negatively impact a person's ability to function and carry out everyday activities. Aside from memory impairment and a disruption in thought patterns, the most common symptoms include emotional problems, difficulties with language, and decreased motivation. And going over here, so transcriptomics technologies are the techniques used to study an organism's transcriptome, the sum of all its RNA transcripts. And going above, yes, most of the above listed functional non-coding RNA deal with regulation of expression. And that's the ones all the way described over here. Going for this, so causal bodies or CBs, and we discussed those over here. We mentioned it briefly. So this scaRNA is small casual bodies specific RNA. So let's go over those. So casual bodies, CBs, are coiled bodies. Also called coiled bodies, are spherical nuclear bodies of 0.3 to 1.0 micrometers, which is that's a millionth of a meter, in diameter, found in the nucleus of proliferative cells like embryonic cells and tumor cells that keep growing, or metabolically active cells like neurons. All right, let's continue further, and now let's look at the updated diagram of the central dogma of molecular biology. So the roles of non-coding RNAs in the central dogma of molecular biology, and so we have ribonucleoproteins are shown in red. So all these are in red, and these are ribonucleoprotein. RNP is a complex of ribonucleic acid, RNA, and RNA binding protein. So basically you'll have the RNAs, non-coding RNAs, plus the protein. Yeah, so in this case, these are all just non-coding RNAs plus protein. And note also the non-coding RNAs are in blue right here without the protein. So this one's a tRNA or transfer RNA. And it says here, note in spliceosome snRNA is used. So we'll have SN, which is small nuclear RNA, and then this SNORNP. That is right over here. That's going to be the SNO is the small nucleolar RNA, and then plus a protein. So that's the RNP. So we get that RNP there. And let's just go over the setup. So what we'll have is mDNA right here. It goes over to pre-mRNA via transcription. And just showing the information flow. So we'll have mDNA. This is labeled, I believe this is going to be just messenger DNA or mature DNA. And it's referring to basically what the RNA is going to be formed. So pre-mRNA, and then that one's that. Then the next one is splicing, and in splicing you have this uDNA. That is this. I couldn't find the exact abbreviation or the relevant definition. See note below. We'll get to that in a bit. So basically, we'll have DNA plus this small nucleolar RNP or SNORNP, and then it produces a spliceosome, and a spliceosome also uses snRNA. They either uses or used in the production of this, so it creates a spliceosome. Then we'll have splicing. So you splice the premature RNA into mature RNA or messenger RNA. And this mRNA also has another ribonucleoprotein RNP right here. So this RISC, and this involves or derived from mRNA. And that mRNA is the microRNA. So right here. Yeah, so it's a small RNA such as microRNAs, miRNA. So I'm not going to go over each one of these RNAs or RNPs individually. I'm just going to illustrate them just to specify that a lot of the information flow from DNA to protein, DNA to RNA to protein involves non-coding RNAs plus proteins connected to them in most of them. So what we'll have is mRNA involves RISC, which is a RNP derived from mRNA. And now the next setup is translation. Yeah, and in this translation, that's via the ribosome, and the ribosome involves these rDNA, which is the ribosomal DNA, and then plus this ribonucleoprotein setup, so RNAs, MRP plus SNORNP, and then it produces the ribosome. So that DNA is used to produce the ribosome via this non-coding RNA inside this protein setup. And then that is involved in the translation. Then we get a nascent protein. And nascent means coming or having recently come into existence. So basically, a new protein is formed. And during this ribosome, remember, it also involves the help of a transfer RNA, and that transfer RNA is produced from a transfer DNA, so tDNA, transfer DNA, plus an RNase P, so again, another RNP, or non-coding RNA, plus the protein gets to this tRNA, then you get this ribosome, plus the ribosome translates the mRNA into a nascent protein. We can see this setup earlier that I went over. Let's just go to transfer RNA. That was where it was. So over here, so a transfer RNA, formerly sRNA for soluble RNA, is basically abbreviated tRNA, is a adapter molecule composed of RNA and it serves as a physical link between mRNA and the amino acid sequence of protein. So you have the ribosome right here, then the transferred RNA grabs the amino acid and then connects over to here and then releases itself, forming a chain of amino acids in the ribosome. So yes, fascinating stuff. All right, so the next step here, this one's an interesting one. I believe this is for bacteria. So this is, you get the nascent protein or the new protein, and then there's a transport one, and this one here is a SRP tmRNA, and you get this localized protein over there. And this tmRNA, I haven't mentioned it or wasn't in the earlier notes. So what I did was I just added it here. So transfer messenger RNA, abbreviated tmRNA, also known as 10saRNA, and by its genetic name SSRA, is a bacterial RNA molecule with dual tRNA-like and messenger RNA-like properties. So yeah, hence you get this extra step there. And this one here, I got this from this nature.com paper. So in bacteria, there's a translation is the main rescue system. So yeah, translation. So you have translated. Now there's a second one, transport, or the translation setup, or the post-translation setup. So translation is the main rescue system, freeing ribosomes stalled on defective messenger RNAs. Yeah, so if you have the ribosome on RNA is defective, so what you're going to do is we'll need to free that up because it's not producing anything. So what we'll do here is keep reading it. So this mechanism is driven by a small protein B called SMPB and transfer messenger RNA, or tmRNA. And this is a hybrid RNA known to have both a tRNA-like and an mRNA-like domain or properties. Yeah, so basically it's like a messenger. It's like a messenger RNA where it produces a protein, and it's like a transfer RNA which can actually transport or function physically. Yeah, and basically moves that protein away. So it's a very interesting setup there. So this one's extra for the bacteria. I believe that's just for the bacteria. But again, I'll have to, this would require another video on its own to fully dive in. But anyways, let's continue further. And now we'll look at that uDNA note. So this one right here, uDNA, just I couldn't find the abbreviation, so see note below. I couldn't find the exact one. I found one that was unintegrated DNA, but that was for HIV, HIV studies, or HIV papers, and also urine DNA, but that's specifically for getting DNA from urine. So those are not relevant. So uDNA note, I couldn't find any relevant definition for uDNA. I am assuming it relates to the different types of small nuclear RNPs that join the spliceosome, yeah, which are labeled as U1, U2, U4, U5, and U6. So it's probably something like that, I would assume. So at least 5 different kinds of snRNPs, this is from Wikipedia now, snRNPs join the spliceosome to participate in splicing. They can be visualized by gel electrophoresis and are known individually as U1, U2, U4, U5, and U6. Their snRNA components are known respectively as U1 sRNA, U2 sRNA, U4 sRNA, U5 sRNA, and U6 sRNA. So yeah, so here we have the uDNA, and I assume it's discussing about the types of the right here of the snRNPs that are formed, or slash snRNA. Yeah, which join the spliceosome. So you have this right here, uDNA gets this snORNP, this ribonuclear protein to form the spliceosome, and spliceosome has a bunch of other, or spliceosome uses the snRNA, and something like that. And I think what I'll do now is actually just copy and paste the SNORN, this one right here, just to go over the definition over here. So in molecular biology, small nucleolar RNAs, or snRNAs, are a class of small RNA molecules that primarily guide chemical modifications of other RNAs, mainly ribosomal RNAs, transfer RNAs, and small nuclear RNAs. So yes, basically it modifies the snRNAs, which are part of the spliceosome. And as well as here, this ribosomal one also modifies that. Yes, there's ribosomal RNAs and transfer RNAs as well. So it can modify it in this as well. Yeah, so basically I think this uDNA is relating to the types of these snRNAs, which are labeled using the U1, U2, U4, U5, U6 labeling methods or terms. So hence this DNA. So that is what I state. But if you know otherwise, let me know, because I can find this anywhere specifically stating that. And also note that gel electrophoresis, that's right here. Yeah, the snRNPs can be visualized by gel electrophoresis. So gel electrophoresis is a method for separation and analysis of macromolecules, DNA, RNA, and proteins, and their fragments based on their size and charge. Interesting. Now let's continue further now. So the genomes of various model organisms, such as Arabidopsis, which is a flowering plant, fruit fly, mice, nematodes, type of worm, and yeast have been sequenced. The sequencing of these are model ones just used. Yeah, we'll get to that in a bit. The sequencing of the entire human genome has yielded practical applications such as DNA fingerprinting, which can be used for paternity testing and forensics. So let me just note, a model organism, often shortened to model, is a non-human species that is extensively studied to understand particular biological phenomena, with the expectation that discoveries made in the model organism will provide insights into the workings of other organisms. And going further here now, so DNA profiling, also called DNA fingerprinting, is a process of determining an individual's DNA characteristics and DNA analysis intended to identify a species rather than an individual. It's called DNA barcoding. Yes, very fascinating stuff here. So human reference genome data by chromosome. So you have the chromosome 1, 2, all the way to X and Y, as well as the mitochondrial DNA. And it shows you the length right here in millimeters. That's a very interesting size there. So you'll have 85 millimeters. Yeah, millimeters is a thousandth of a meter. Yeah, note the lengths here. So 85 millimeters, pretty long. That's 8.5 centimeters, but then again, it's extremely jumbled up. And going further, so mitochondrial DNA is extremely small. This is 0.0054 millimeters. That's very small. Then there's base pairs, 248 million for the chromosome 1, and then you have the mitochondrial, just 16,000. Very interesting. And then variations in these, so different between different humans and so on. You can have variations like that. So 12 million difference between this other one. And then protein coding genes, pseudogenes. We'll get to those in a bit. The next one is going to be total long non-coding RNA, total small non-coding RNA, then microRNA, RNA, snRNA, snORNA, then miscellaneous non-coding RNAs. You have a whole bunch of those. But these are in the thousands versus the total, base pairs. No, this is just total. And then the protein coding ones is in the, also 20,000 there. So there's more, looks like there's more non-coding RNAs. Yeah, so there's more non-coding RNAs, and there's some pseudogenes that are in between there. So pseudogenes are non-functional segments of DNA that resemble functional genes. Most arise as superfluous copies of functional genes, either directly by DNA duplication or indirectly by reverse transcription of an mRNA transcript. Pseudogenes are non-functional segments of DNA that resemble functional genes. So they look like them, but they're not. And it could be just a defective one or superfluous or extra copies of functional genes. Most arise as superfluous copies of functional genes, either directly. Actually, no, I think that's just a copy and paste exact same sentence. I'm just going to delete that. So pseudogenes are usually identified when genome sequence analysis finds gene-like sequences that lack regulatory sequences needed for transcription or translation, whose coding sequences are obviously defective due to frameshifts or premature stop codons. So a frameshift mutation, also called a framing error or a reading frameshift, is a genetic mutation caused by indels or insertions or deletions of a number of nucleotides in a DNA sequence that is not divisible by 3. And this is due to the triplet nature of gene expression by codons. The insertion or deletion can change the reading frame, the grouping of the codons, resulting in a completely different translation from the original. So you have a non divided by 3 because it's the triplet nature of gene expression. It's in by codon. So a stop codon or termination codon is a codon nucleotide triplet with messenger RNA that signals the termination of the translation process of the current protein, so signals when it is. Basically, it's like it's like a recording film for a camera, and there's a defective error in between. It just stops the full video or full image from being shown, which is quite interesting. It's like so DNA is like a programming sequence or film tape, etc. So in medicine, sequencing of the entire human genome has allowed for the identification of mutations that cause tumors, as well as genes that cause a specific genetic disorder. MES note, a mutation is an alteration in the nucleotide sequence of the genome of an organism, virus, or extrachromosomal DNA. Mutations are a result from errors during DNA or viral replication, mitosis or meiosis, or other types of damage to DNA, such as by exposure to ultraviolet radiation, which then may undergo error-prone repair, cause an error during other forms of repair, or cause an error during replication called translesion synthesis. Mutations may also result from insertion or deletion of segments of DNA due to mobile genetic elements. Yeah, so basically mutations is an alteration of the nucleotide sequence of the genome. Going further, there's several different ways, either from damage or just even random chance. Yeah, so going further, so mobile genetic elements, MGEs, sometimes called selfish genetic elements, are a type of genetic material that can move around within a genome, or that can be transferred from one species or replicon to another. MGEs are found in all organisms. In humans, approximately 50% of the genome is thought to be MGEs. Yeah, so which is very interesting. So basically, you can have segments of DNA that's moving around freely or semi-freely, and then they can move around within a genome, where that can be transferred from one species or replicon to another. And yeah, so basically it results, inserts or deletes from a segment. So again, that random chance factor. So a replicon is a DNA molecule or RNA molecule or a region of DNA or RNA that replicates from a single point of origin. So it just keeps replicating from that single point, and then you have these MGs just floating around. And approximately so MGEs are found in all organisms. In humans, approximately 50% of the genome is thought to be MGEs that are selfish genetic elements that can move around within a genome. This raises a lot of questions about the actual central dogma theory. And if you think about it also by viruses, once you look into them, they're pretty much just a lump of genetic material plus proteins, and they move around. In other words, you have a mobile genetic element. A replicon, yes, we went over that. The origin of replication right here, also called the replication origin, is a particular sequence in a genome at which replication is initiated. And a genetic disorder is a health problem caused by one or more abnormalities in the genome.